Hello, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Quality Pillars for Building Measurement Assurance in Flow Cytometry, and our sponsor is Streck. Our panelist today is Dr. Virginia Litwin, Vice President of Immunology at Caprian Biosciences. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the control panel to ask a question. Please make sure the drop-down menu says All Panelists. If you do not see the Q&A box in the control panel, move your cursor over the main panel until you see the control icons on the bottom. Select the Options menu and then select Q&A. We will ask the panelists questions after the presentation has concluded. Dr. Litwin, please go ahead.
Welcome, everyone, and thank you for attending the webinar today. And I want to thank Streck for sponsoring the webinar, and Denise in particular for asking me to participate today. Our webinar today will have three aspects. We're going to talk about quality pillars, why we need them, what they are, and then we're going to look at some quality pillar process maps and case studies. So why do we need quality pillars? Well, the big spoiler alert is that flow cytometry has an increased role now in drug discovery and development, and it's being used for more important purposes. So we need to up our game a bit in how we perform flow cytometry to support drug discovery and development. So you'll all recall that about 20 years now, biomarkers, or the translational approach to drug development, was applied. And the purpose of using biomarkers and this translational approach was to increase the success rate and decrease the timelines of bringing new therapeutics to market, to allow for more informed decision-making, and to build the therapeutic potential and drug labeling claims. So overall, biomarkers fell into two main categories: scientific advancement and internal decision-making, or regulatory submission. So enter onto the scene maybe 10 years ago, the new investigational immunotherapies. And because of them, the new vaccines and the new cellular therapies, flow cytometry has become even more important in drug discovery and development. So immunotherapies are a novel class of compound, and their mechanism is to harness the power of the immune system. They might rev it up and they might tamp it down, or they might deplete a targeted cell population altogether. Immunotherapies come in all shapes and sizes. There are small molecules, biologics, or cell-based therapies. Mostly they're used in oncology, for inflammatory diseases, as well as autoimmunity. And I know everyone online is as delighted as I was to see that two of our pioneers for immunotherapy, Dr. Jim Allison and Tasuku Honjo, were awarded the Nobel Prize this year for their role in not only identifying the mechanism of action of CTLA-4 and PD-1, but figuring out how to use those compounds to harness those compounds for new cancer therapies. So we can't congratulate them often enough. So if we look then for the role of flow cytometry in drug discovery and development, for each of these areas, immunotherapeutics, vaccines, and cell therapies, it's become more important. So for investigational therapies, in the drug discovery phase, flow is being used for target ID, specificity, and mechanism of action. In the development phase, when we give the drugs to patients, we're using flow cytometry to measure immune status, evaluate targeted cell depletion, evaluate minimal residual disease and leukemia lymphoma. And of course, when we have a protein-based therapeutic binding a molecule on the cell surface, flow cytometry is the best technology to measure receptor occupancy. For novel vaccines, we use flow in discovery phase for proof of concept, mechanism of action, and in the development phase, when we've given the vaccine to a patient, to measure their overall immune status, as well as for efficacy. We can measure the immune response to the vaccine through intracellular staining or by monitoring antigen-specific T cells by staining with multimers. And of course, with cellular therapies and regenerative medicine, Now, all of a sudden, flow cytometry is part of the manufacturing process because the drug is, in fact, a cell. We use flow cytometry to look at the viability, purity, quality, identity, and stability of the drug product. And once the drug is administered, we use flow cytometry for pharmacokinetic analysis, looking at the viability and stability of the cells in vivo, as well as their identity. Have they done any phenotypic changes on us? Because of this increased importance of flow cytometry in drug development, this is why we need our quality pillars for measurement assurance. So just to summarize, we're now using flow cytometry in the manufacturing phase, and this is under GMP requirements, in preclinical testing for toxicology, for lot release of these various biologics, so that would fall under GLP, and no longer just in clinical development as an exploratory or secondary endpoint, but for a primary endpoint, and as I mentioned, for PK. So these now fall under GLP. And you'll notice that I have here good documentation practices under all of these. And in addition to how we run our assays, we need to make sure that we now document everything appropriately. So this is why we need quality pillars. And in addition to quality pillars, we need measurement assurance. And we also need physical standards and guidance documents for how to conduct flow cytometry in a regulated environment. And these last 2 points, I will give you a brief update at the end of the webinar. So what are quality pillars? Well, the foundation of quality pillars are quality management systems and measurement assurance. So quality management systems, please do not log off at seeing this slide. It's not a QA talk, but I do need to introduce everyone to some QA concepts. So quality management systems are required in regulated labs and manufacturing, as well as other industries. So the objective is to reduce or eliminate errors, meet customer expectations, realize a more efficient and effective operation, meet regulatory and accreditation requirements, and meet your institutional quality objectives. Now, in some cases, the institution has higher quality standards than the regulatory, and that's great, but they can never be lower. So then your quality management system needs to be aligned with the good old-fashioned quality systems essentials. And those are the 12 building blocks of quality. So there are aspects that will look at your organization, your process management, your customer focus, documents and records, facilities and safety, information management, personnel, non-conforming event management, purchase and inventory, assessment, equipment, and continual improvement. There's not a test at the end, but if there were, you'll see that the presentation today focuses on most of these areas. So measurement assurance, this might be a new phrase for some of you, and so I just have this one slide to introduce you. And the concepts from this slide I took from a presentation by John Elliott at the NIST FDA workshop, which was held in 2017. I have a link to that workshop, and there's some more links at the end. So if you're interested, you can download all the presentations from that workshop, and there was quite a bit of interesting information. So of course, at NIST, the National Institute of Standards and Technology, their business is measurement science. And they define this as a systematic approach that informs on the comparability and confidence in the result. And this allows for data-driven decision-making. So in Dr. Elliott's presentation, he talked about 3 components of a measurement. And I just thought this was interesting and a different way to thinking about our data. So first of all, we have our value. Is it on scale? And if it's on scale, then I'm not sure if you guys can see my pointer or not, but if it's on scale, you can then compare your value to another value. Uncertainty is the variability in the measurement, and this lets us know how reliable that measurement is. And the variability of the measurement we establish during validation. And then there's evidence, evaluation of your measurement system, or how much confidence can you have in that measurement. And this is what we're going to talk about today is our quality systems give us confidence in our measurement. And then he had a bullet point in his presentation, which I really liked, and he said to treat your assay as the measurement process, and that's what we're going to do today. So the building quality pillars for measurement assurance, what do we need to do to build our pillars? So first of all, we need to define the elements that contribute to our measurement. And then for each of those elements, we need to define a process, because we need to establish and maintain a controlled environment so that we can produce reliable data over a long period of time. We need to define performance indicators for each of those elements. And we need to define how to maintain and monitor the performance indicators. So what we want to do is manage the life cycle of our process. This isn't one of those set it and forget it processes. So we need to define our key performance specifications, so our acceptability criteria. So let's do that for flow cytometry. So let's build our quality pillars for measurement assurance and flow cytometry. So the elements of the measurement I've defined as these 5 things here, and those will be our quality pillars for flow cytometry. Our instrument, of course, our method, how we report our data, how we monitor, and how we document. So in this presentation today, I'm not going to go into a lot of detail on any one thing, but I want to give you a little highlight and talk about some process maps and some case studies for each of these 5 quality pillars. So let's start with our instrumentation. So there's each quality pillar has some sub-pillars. So for the instrument, our sub-pillars are the qualification, our optimization and characterization, our daily setup and assay specific settings, and instrument standardization. So the instrumentation quality pillars provide assurance that the data are generated from an instrument that's reliable and precise. And this is the foundation of good data. So for instrument qualification, I don't have time to talk about it today, but Cherie Green just gave an excellent deep dive webinar from ISAC at the CytoU, and there's a link for it here. And I apologize, but I will be pointing you to lots of other webinars today, and you can look at them or not, but just so you get some, if you want a little more information on any topic. And Cherie's webinar was part of a 3-part webinar series that we put together for ISAC. So let's talk about instrument standardization. Why standardize? Well, instrument variation is a major source of variability in flow cytometry. You can have it within your lab, between experiments, and when we go to multi-center clinical trials, it's a huge source of variation. So this is something that we want to control. The goal of instrument standardization is simple: to reproducibly set your gains to achieve equivalent fluorescence experiment to experiment, so that could be on the same machine, instrument to instrument, lab to lab, or even platform to platform. It also allows you to accurately assign your spillover values for compensation. Compensation is also a major source of variation in the data. It allows you to maintain consistent longitudinal fluorescent measurements. I'm not going to discuss today any one process of cross-standardizing your instruments. There's many different ways you can do it, but I do want to show you a case study. So here we've cross-standardized 2 instruments, and for instrument monitoring, what we did is we took the comp beads stained with the antibodies from each of the fluorochromes used in our assay, and we then measured those on each instrument after we had applied the application settings and all of that. So we measured then true fluorescence, and we calculated the % difference between the 2 instruments, as well as the % CV. And you can see that the results are very impressive. There's less than a 4% difference and less than a 3% coefficient of variation between the fluorescent output that we get between these 2 instruments. And I can tell you, I've done this exercise with instruments that were not standardized. And in this case, the instruments were fine. They passed their daily CSNT. There were no issues at all, but they weren't cross-standardized. And in some of the detectors, not all, but we saw as high as 85% CV. So if you can bring that down to under 3%, this is time well spent. Now, of course, in flow cytometry, the proof of the pudding is in the plots. And so here you can see that we've run some samples on each of these instruments, and the plots from both instruments are nearly identical. So we get the same mean fluorescent value and the same nice separation between our positive, negatives, and DIMs. So now let's talk about the method quality pillar. So the sub-pillars for the method quality are the development and optimization of high-quality methods. If you don't develop a good method, you're not going to get good data in the end. Assay characterization and validation. And then you need a process maps for your pre-examination and the examination phase of your assay. So when we develop assays for drug development, it's a little bit more rigorous than when we do that in other environments. So we need to fully understand the clinical question that is being addressed by this method and before we start anything. And what decisions will the data enable? What will the regulatory oversight be? And so this information must guide our decisions regarding the assay matrix as well as our performance requirements. So the process map overall from assay development to validation is as follows. You would design your assay, and then you want to optimize it. So you want to determine the most favorable conditions for running that assay, and you want to stress test it. Once you've done that, you want to do a preliminary characterization, look at your precision and your stability, and then ask the question, are these data acceptable for the intended use? If yes, then you get to go ahead and go into validation. If no, then you need to go back and re-optimize or maybe even redesign the assay altogether. Now for validation, 2 of the webinars I mentioned earlier have gone into a deep dive on validation, so we don't need to address that today, but they're available for download if you're interested. So let's look at our assay options. So our assay types, what do we want to do? In clinical labs as well as in drug development, mainly we look at immunophenotyping by monoclonal antibodies or multimers, leukemia lymphoma assays, minimal residual disease, phospho receptor occupancy, or functional assays, as I mentioned, intracellular staining. We also are now doing pharmacokinetics. We're looking for those cellular drugs. Our data output would be the relative percentage of the parent population, cell concentration, fluorescence intensity, percent bound or free in a receptor occupancy, or in leukemia lymphomas, a phenotypic description of the abnormal cells. Our matrix mostly is whole blood, bone marrow, PBMCs or BBMCs, and tumor-infiltrating lymphocytes, as well as a few others. So we're going to develop our assay. Let's establish the assay objectives. What do we want to measure, and in what context, what matrix? And then we're going to decide how we want to define our population of interest. What will be our positive and negative selection antigens? And I have throughout this assay specificity thrown in here and there, because everything we do from developing the assay contributes to our assay specificity. So that's a way where flow cytometry is different from other technologies. Okay, so we know what we want to do. Now we need to look at what instrument we have, of course. What are the number and type of lasers, the number of detectors, and our filter configuration. When we have that information, we can design our panel. We can pick the appropriate fluorochromes for the appropriate monoclonal antibodies, and we would want to do a monoclonal antibody clone evaluation to make sure we've got the most specific clone available. We titrate our reagents, and then we figure out the appropriate wash, lye, sticks, sequence, and we evaluate different buffers if we're looking at intracellular staining. Again, this greatly influences assay specificity. Now we can design our templates for acquisition and analysis. And obviously our data analysis templates We need to ensure that the population of interest is within our gate, and other things are not. And this contributes to assay specificity. So in a regulated environment, we're talking about the method quality pillar right now. We need a process map for creating those templates. And what we want to see in a template should be defined in an SOP or another driving document. So there's requirements that you will have at your institution that you need to have in that gating and acquisition template. And then you need to have the template reviewed by someone else in the software with some data. You need to stress test that. So, sorry, I'm hearing an echo back. So if Genome Web can adjust that, that would be great.
I'm sorry, Virginia, there's nothing we can do about that.
Okay. Well, hopefully no one else hears it. Okay, anyway, so we've stress tested our templates, and now we need to approve it. And I mentioned that documentation was one of the quality pillars, so we need to document that approval process and then lock it down with setting our access permissions and all of that. And we would need to do version control on the template. Okay, so now we're going to optimize our method. And this is not an exhaustive list. There's many things we can do during assay optimization. We do want to look at our acquisition templates and see what we should put the threshold parameters, how many number of events we should acquire, things like that. We also need to look at our shipping conditions and our storage conditions. So we really want to stress test that method before we go into validation. So here's a case study where we looked at whole blood collected in cytocheck tubes, and we compared shipping and storage at room temp versus 4 degrees. So on day 0, you can see everything looks quite nice, but over the subsequent days, we found that shipping and storage at 4 degrees, we saw more optimal red blood cell lysis. And so part of this assay optimization was to choose that as our shipping and storage conditions. So let's talk about specificity a little bit. And I think you verified specificity more than validated. But in this, for flow cytometry, I think of specificity in 2 things. Are my reagents specific, so the monoclonal antibodies, and am I reporting what I think I am? So are the cells in my gate what they think they are? So I have a very simple example here. And if you look at the flow plots, again, I'm not sure if you can see my pointer, we've gated on forward and side scatter, and we've been a bit sloppy here on purpose. So we've included lymphocytes and monocytes. And then when we look to the plot to the right, we want to look at our CD4 T cells. And we can see them. We also see another population in that gate, which are not T cells, they're in fact monocytes. So they're dim CD4. So this gating is not specific. We go down on the bottom, it's done properly. We have an appropriate gate on the lymphocytes, and we see them when we look at CD4 versus CD8. Yes, we have a specific assay for CD4 T cells in this case. Other things we do to verify specificity could be blocking reagents, biological positive and negative controls. In terms of what marker we're selecting, we can look to the literature and make sure that we have the correct phenotype for that subpopulation. We can do ex vivo stimulation to induce activation markers. Leukemia, lymphoma, we would absolutely need to use disease-specific samples. And now in the age of immunotherapeutics, when our drug could be an antibody that is present in our panel, we need to either exclude that CD marker from the panel or find an alternative or prove that they don't cross-compete. And examples are CD19, CD20, CD38. So we need to show that our assay is specific. So here's a simple case study of verification for specificity. we have intracellular cytokines, and certainly our FMO controls in the assay are a way to demonstrate specificity. So on the top panel, the FMO, we have no events in the gate for these three cytokines. Then we look at unstimulated cells. We might see a little bit of background, maybe some endogenous activation, but nothing much. And then we do a serious activation with SEB, and we can see, yes, indeed, we have a nice control here where we have upregulated the expression of these cytokines. And then on the bottom, it's more of a disease state verification where we have confirmed CMV positive donors, and we've stimulated ex vivo with CMV, and we see a more biological response of antigen-specific cytokine production. So there's lots of ways we can verify specificity. Now let's look at a process map for the examination phase. So we're going to test our sample. What do we need to do? And we should have a process map or a series of SOPs defining this. So first of all, our instrument setup. We need to make sure that our instrument has passed the daily hoops that we put it through. And these things highlighted in red, I'll show you examples of later. Reagent verification. First of all, you need to make sure that the bottle you've got in your hand is the right one, and it's the right reagent as listed in your SOP, that it's within its expiry date, and that it is past the lot-to-lot qualification. Now we're going to look at our specimen and look at, is it the right specimen? Verify the specimen ID. Is it the right matrix in the right tube? Is it within stability? We're going to look at specimen quality. Is it clotted? Is it hemolyzed? Can we use that sample? Now we are ready to go ahead and stain our sample for the SOP. We need to make sure we have the correct acquisition template, the correct instrument settings, and compensation all applied. And now we can hit acquire on the instrument. Next, we're going to process map the post-examination phase, or the run, review, and acceptance. So we need to look at a quick look-see at the data, make sure all the antibodies were added and the staining is as expected. If we have a minimum number of events we need to acquire, like we do with an MRD assay, we need to make sure that we have acquired the minimum number of events. And then we're going to go to the gating phase. We will look at did the files move to the correct location, all the appropriate keywords and everything imported. Are we using the correct gating template? When we've verified all that, then we can go ahead and gate the specimens and the QCs. The QC evaluation, we will look at the QCs. Are they within range? And we will monitor QC performance, and typically that's done in a Levy-Jennings chart. So then if all that goes well, we can approve, release the data, and then do the appropriate archiving and documentation. So now let's talk about monitoring. So monitoring is the overall quality processes that we have in the laboratory. And so quality control is a set of procedures for monitoring laboratory work. It provides evidence that the measurement process is proceeding as expected. And so don't forget, we're going to treat our assay as a measurement process. And so we want to create a quality control process map. And first of all, we want to define what is it we want to control. So we need to ask, well, where does the variability come from? And then we want to define how to control it. What process controls do we need? What QC material can we use? What are the acceptability criteria? And how will we document this? So this is the lovely figure from Holden-Macker's Nature review paper that many of us have used in our presentations. And he's shown the whole process from collecting a specimen from a patient to the final reportable result. And so we can use this lovely figure to ask ourselves, where does variability come from? And the answer is simply yes. Every single step here can introduce variability. So how do we control it? We control it, of course, with our flow cytometry quality of pillars approach. So let's talk about QC material for flow cytometry. And in any laboratory, regulated or non-regulated, if you use QC material in every run, it's advantageous. It really can tell you a lot about your assay. But when you're considering longitudinal data and multi-center clinical trials, QC samples are critical. So the objective of a QC is to ensure that the reagents and assays are functioning properly. So these are assay process controls. I'm not talking about all the beads and fun things that we'll use to make sure that our instruments are behaving. We're just talking about assays here. And so we want to monitor assay performance trending. So as with everything we do in flow cytometry, there's lots of challenges, and there's not a perfect solution for QC material. For most assays, there is no true reference material. The desired characteristics of our QC material is that it's similar to the specimen matrix, and it has a long shelf life. Well, these are almost mutually exclusive. And I want to point you to a nice resource that's in press for control cells for flow cytometry. And at CYTO 2018, there was a workshop on control cells, and all of the workshops from 2018 this year will be published in a special issue of Cytometry A, including this one, and so that's in press for a future resource. Some of the commercially available QC materials, this is pretty comprehensive. There may be some others out there, but there's, of course, preserved whole blood. Now, this is the ideal QC material if you have an assay that's using whole blood or bone marrow. It will allow you to control every step, including RBC lysis. You can also look at lyophilized lymphocytes, which have some advantages. Dried leukocytes, again, these have advantages of longer stability, but they are slightly a different matrix from our cells. But you can see with the dried leukocytes, they're pretty cool. You can resolve even basophils and dendritic cells quite nicely. There are also really clever things, these cellular mimics, which are polymer-based, and you can see that they, for light scatter at least, they function nicely to mimic the lymphs, monos, and granulocytes. So let's look at a few case studies in assay monitoring. So this slide was kindly provided by Sherry Green, and what she's done here is she is looking at an in vitro diagnostic TB&K assay for whole blood lysozyme wash method. So she's showing us two QCs, and many clinical laboratories will use a normal donor every day as the QC matrix, and this is identical specimen matrix. Another approach is to use a commercial control, such as CD checks, and this is preserved blood, and so it approximates the specimen matrix. So let's look at the data, and you can see that the blue is the CD checks, and you have not only tighter CVs, but with the CD checks, now you're only looking at day-to-day performance of the assay. There's no impact, as long as this is used within the stability window, of changes in the QC material. Whereas when we use a normal donor, okay, we can use it to evaluate a lot of our assays, but we also are confounding with day-to-day variation from the same donor and between donor variability. So in this case, it's really better to use a preserved commercial QC. Now here we have the assay is on frozen PBMCs. So the ideal control here is what we did was we banked a large leukopak of healthy donor PBMCs to run each time we run the assay. Now the nice thing about this control is it, again, follows the entire process. The sample gets thawed and along with the patient sample. So if there's an issue with thawing, we'll pick it up with the QC. When we review our data, we should not only look at the Levy-Jennings plots, but we need to look at the flow plots as well. So when we look here at run 1 and run 23, you can see those look quite nice. There's no apparent issue with this assay. And when we look at the Levy-Jennings plot, we can see that it's really tight. There's only a 2% CV, and all the runs pass. The data are very reliable. We can go ahead and report the data. So now we can say with confidence, if we have a patient sample that was here one day and it's way down here the other, we can say, yeah, we're pretty sure that's something with the patient and it's not something with the assay. There's a similar type of assay, frozen PBMCs, healthy donor control. So let's look first at run 1 and run 17. At a glance, they both look nice to me. Same level of fluorescence, good separation. Now let's look at the Levy-Jennings. And we see that the last 4 runs here have failed. And when we look at our overall CVs, there's lots of information we can get out of the Levy-Jennings. Over here, the assay is giving us a 6% CV, which is very nice. And if we include everything, we say, woo, it's 33%. So we know that something happened in these 4 runs, and those data are not reliable and they can't be reported. So let's also look at reagent monitoring. So our lot-to-lot evaluation, which is something we need to do and document. So here we've got a CD3 antibody. We get identical results with the current lot and the new lot. And even if you look down at the MFIs, they look identical. So that's great. That reagent can be put into use. And here we have a PD-1 antibody where this antibody cannot be put into use. There's no staining here. And so we know that lot of reagent is unacceptable. So this is why we go through all the trouble that we do for quality monitoring. Okay, so let's talk about instrument monitoring now. And I'd like to introduce you to Lucy and Ethel. And they, of course, are 2 LSR2 Fortessas, 5 lasers. And Ethel's pretty reasonable. So what I'm showing you here is 1 month's worth of Levy-Jennings data, the daily CSNT, and a plot for each of the 5 lasers. Acceptance criteria is, for the robust CV is 6%, and you can see, well, maybe you can't, but the numbers here are very, very small. So very, very tight. It's really under 4 or 3% variation. Okay, so Ethel's a steady Eddie. Now Lucy's a little bit all over the place. So you can see that she might be pretty consistent, but she might be off. And so, again, this is why we monitor and we document. So here, actually, we're within the 6%, but it's so far different from normal Lucy. So we'll do an investigation there and not use the instrument, but use when it's here. Here, if we've exceeded 6%, we will document that we had an issue and what we did. We realigned the lasers. So this is really important. So you can show then to an auditor, you can prove that, yeah, Lucy was not used unless the CSNT were within the acceptability criteria ever. You can easily document that, reconstruct the data. And you can also use these data to show your service engineer, and that will give the service engineer a lot of information to help diagnose what might be the issue with Lucy and get her on the right path. Okay, so let's talk about documentation. This is a short one, so don't worry. Basically, in a regulated lab, and really in the best research labs, you document what you do. If you didn't document it in a regulatory setting, it didn't happen. And like the overall quality practices, you need to follow the documentation practices that are appropriate to the regulatory requirements of the intended use of your data. And you need to also follow your institution's practices. So that's all we have on documentation. So let's talk a little bit now about guidance documents and standards. So I'm going to start with this slide. Can I do that? Yes. So the NIST and the FDA last year hosted 3 workshops on cell counting, flow cytometry, and genome editing. And I actually attended one of these workshops, but and the reason they had them was because of this 21st Century Cures Act, which I was unaware of existed until I attended the workshop. So within this act, there is a regenerative medicine provision, and that mandates that Health and Human Services, in consultation with NIST and stakeholders, facilitate efforts around the development of standards for regenerative medicine therapies. And you can go to the link here. So what NIST did is, along with the FDA, their action items to fulfill this mandate, one of their action items was to have these 3 workshops. And I've put the links here. So one of the outcomes of the workshop on cell counting is there's now an ISO document on cell counting, which you can look at here. And there's also a reference standards initiative from NIST. And I think many of you have heard Lily Wang speak about this, and her slide deck is available at this link here. So we are looking for ways to make sure that reliable data are generated. And there should be an outcome for the flow cytometry workshop as well. So the other area is validation. So all of us working in clinical laboratories as well as in drug development and the manufacturers, we would love to have guidance documents for the validation of flow cytometric methods. And we don't have any official guidance documents yet. Therefore, many of us from academia, biopharma, CROs, clinical labs, reagent manufacturers, and government agencies submitted an application to the Clinical Laboratory Standards Institute for a guidance document. And I have here a link to a detailed presentation on this document if you're interested. And so the hope is that this guidance document will come out and then it will be recognized by the regulatory agencies. But first we have to get it finished. So the process and timelines for the guidance document are as follows. At the end of this month, we will submit the document to the writing committee, will submit the document to the CLSI. And then in February, this is the important part, it will be posted on the CLSI website for open comment for 60 days. So we hope everyone comments on the document and provides their feedback. Then we have until June to respond to each and every comment. And if you're not satisfied with how we have responded to your comment, there is a 30-day appeal period. And so then finally, it will go back to CLSI in August, and it will be published in December of next year. And this is part of why the CLSI document carry more weight than our typical white papers. And first of all, we have, the CLSI has rules on the composition of the voting members of the committee, and we needed people from industry, professionals, as well as government reps, which are the NIST and the FDA. And the other reason that the CLSI document carries more weight is this review process. So we really hope lots of people comment on the document. And so just to summarize now, why do we need quality pillars? Well, it provides assurance that the data are reliable and precise, and we need to ensure integrity and reconstruction of the data. So all the quality pillars will allow us to do this. And then the take-home message is to sweat the small stuff. In flow cytometry, it's all small. It's less than 70 microns. And the devil is truly, truly in the details. So thank you so much. And I'd like to open up at this point if anyone has any questions. And also my contact information is here if you want to contact me later. That's fine.
Thank you, Dr. Litwin. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. All right, we'll now start the Q&A. Virginia, for gating templates for assays to be used in the clinic or in drug development, are those ever generated using automatic clustering algorithms, or are those always manually locked down?
Sure. There are, so yes, you can use those, but you need to validate them. And there's some very impressive new softwares out and processes that generate some interesting data. They need to be fully validated, and I think they're great. They save time and give us more consistent data. But we need to also understand them. So I'm always a little hesitant to put something in my validation that I don't understand pretty well. So, we can't have a black box kind of thing. But yeah, those are, but you would need to then in your documentation describe, okay, the algorithm and how it's being used, what decisions does it make for the gating.
Okay, great. And our next question is in regards to quality control processes for targeted phenotypes that aren't covered by readily available control cell materials, such as CD4+ or IL-17+. And the question is, do you still find it useful to utilize controls that only cover the higher up lineage markers or phenotypes, such as CD4+?
Well, okay, that's a great question. So with the control material, there are, okay, long question. So we don't have standards, right? We don't have a reference material that's going to have, okay, my assay has 23 reportable results. No, you will not find, you won't get target values for all those in your reference material. But I've found that mostly you'll find the population and you can monitor it in your reference material, and that does add value, especially if you've got a traditional gating algorithm. So, and in some cases, you can't, very few cases have I seen where I get such bad CVs that I can't monitor that population with that QC material. But what I'll do then is I'll just make sure that I have a population, say with PD1, where I do have PD1 reasonably expressed, and I can monitor that population.
Okay, great. You spoke about standardized instruments. Are those standardized by different companies, company by company, or are there any common criteria for those standards?
Right. So there's different ways you can do it. There's publications on it. I've worked with one instrument vendor to cross-standardize the instrument, but many vendors have a process to do that. So that it's, yeah. But there are also ways where you can do it if you're very clever between vendor different platforms. And I think in the webinar from Sherry Green, she does address this. It's a long answer.
Oh no, please continue if there's anything else you wanted to add.
No, I mean basically the bottom line is you're going to try to have your instrument so that you can get a target value on all the instruments in the same space every time, the same place. So, yeah, I would just refer you to the publication, sorry.
Okay. And sort of continuing along that same line, can you tell us what's the best way to control for instrument variability even though they're all the same instrument platform?
Is that between or within?
Both.
Okay. So my experience with the newer instruments is they're very stable. And the between instrument variability, again, just cross-standardize your instruments. And also we learned a bit the hard way, but I mentioned it briefly here. If you monitor with hard dyed beads, that's going to add some variability as well. So when you do your standardization and your monitoring, you want to use true fluorescent beads. So comp beads work, but now you've got an issue of we're making a prep every day and I've got different antibodies or different lots. So if you can use something that's more controlled, like a lyophilized fluorescent bead or something, you can push to all your sites. That's the best approach.
Okay. Going back to the quality requirements that you were talking about, have those already been implemented, or is this a plan for implementation? Can you talk a little bit more about that?
The quality requirements I talked here, the quality pillars, are industry standards, really, things that we've seen from other publications and good QA practices that I just wanted to highlight those 5 areas. For the CLSI document, if that's what you're referring to, that will be published in a year from now.
Okay, great. When performing stress tests for storage or shipping conditions, how many samples do you think should be tested at each condition?
Good question. Personally, I like 3 to 6. There aren't really rules around that. And in flow cytometry, we have to bear in mind that it's expensive. And so we don't want to get too crazy with our experiments. A minimum of 3, because we will be looking at donor-to-donor variability. But I usually find that 6 is sufficient as well.
And what would you say is an acceptable % change in reagent lot-to-lot evaluation?
Okay. So when in the validation webinar that I talked about, I made the point of, in my opinion, within assay precision is the most important thing. It's really important to validate that. So you need to understand is when I take a sample and I run it 3 times in an assay, are my CVs 2% or are they 20%? So if my inherent variability is 20%, when I look at lot-to-lot or stability, I need to keep that in mind. And so when you talk about lot-to-lot, you need to keep in mind what's the variability of staining within the assay as well. So if you get a high variability in your staining, then you need to allow for that when you look from an old lot to a new lot.
Great, thank you very much. And those are all the questions that we have in our queue. We'd like to thank Virginia Litwin and our sponsor, Streck. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archive version will be emailed to all attendees. Thank you for joining us for this genome webinar.
Thank you.